essentially, if I had just used the model and said, I'm going to take the 10 outputs from this model and test them in my system right away, I could have saved myself about 6 months of work. Yeah, my name's Sam. I'm a third year PhD student in Peter Jackson's lab. Project is focused on kind of identifying locally secreted factors that control de novo adipogenesis. So we want to understand how these two cell populations are talking to each other, specifically how the endothelial cells are informing pre-adipocyte fate and kind of regulating adipose tissue expansion. And so one assay to understand before I start to get into any data is this in vitro differentiation assay, adipogenesis assay. Basically, you can take pre-adipocytes, whether that be a cell line or primary isolated pre-adipocytes, culture them up to confluence, stimulate them with this cocktail of drugs that induces differentiation, insulin, IBMX, which raises cyclic AMP, and then dexamethasone, which is a synthetic glucocorticoid. And over the course of 8 days, you can see them literally before your eyes transform into these nice round adipocytes. And the kind of twist that I put on this assay was to collect conditioned medium from endothelial cells. At this point, what I have basically is a big list of proteins. I know which proteins are secreted by these endothelial cells, and we want to know which ones cause activity. So this is kind of our traditional pipeline where we identify what proteins are there, apply them, and trace activity. I at this point was informed of Cellformatica, reached out to Yuri, and basically what I gave him was, this is our data that comes out of our mass spec, which is just a list of the relative abundances of the proteins secreted by endothelial cells. And I gave Yuri the list of the top, I think, 100 most abundant proteins, and had him run them through Cellformatica to get to get the model's idea of which proteins it thinks may be causing this activity. I included, a guiding question, like Yuri mentioned. I included a list of key terms, and then we fed it this list of 100 proteins. And I think Yuri ran each one through about 5 times. So I was given back this list of outputs that looks like this. And there were about 500 outputs. So we have the gene symbol, the protein symbol, the effect size. So this is the predicted effect size from 1 to 10 that Cellformatica predicts this protein will have in my system, a confidence score, so how confident it is in that predicted effect size, and then a little description of how it thinks that particular protein could be acting in my system. And so at this point, I had a list of proteins that I was kind of already interested in, having done my own literature search. And so basically, initially what I did was just take this huge output, kind of skim through it to look at the ones I was interested in, see what the predicted effect size was, and then continue along my way with the biochemical techniques that we've traditionally used in our lab. But I came out with this one target, pentraxin 3, which seemed to be present, secreted by the endothelial cells, and present in both of the fractions that had activity. By early February, mid-February, using traditional biochemical techniques, I was able to identify this target, which has the activity that we're interested in. And I thought at this point, let me just go back to the Cellformatica output and see where this target was in its list of outputs. And it turns out it was actually the number 1 predicted target to have an effect. Number 1, and then also, like I said, I think Yuri ran each target that I gave him through 5 times. So it was 5 of the top 100 outputs were this pentraxin 3. So essentially, if I had just used the model and said, I'm going to take the 10 outputs from this model and test them in my system right away, I could have saved myself about 6 months of work. And it's good to have kind of these orthogonal methodated it biochemically. But clearly I was stunned by this, and it really boosted my confidence that this language model is actually doing something really potentially helpful to researchers, and could have cut a lot of work out for me had I just looked at the outputs and utilized them right away.